talk about why research is important. I can provide you all pictures and diagrams and descriptions and recipes and DNA and RNA and all that, but Jason, after 5 or 10 minutes, has you hooked. He's a great guy, and it's great to meet these guys here as well as you all as we go forward. So next, Dr. Sabrina Cote. Sabrina, so I was contacted by Sabrina's former postdoc advisor who's at Duke, and he said, I've got a very smart young woman who's moving down to Charlotte. Do you think you have a job for her? Because he knew I directed the genomics core here, and I said, well, what does she work on? And he said, well, she works on yeast, the stuff that brews your beer, and the chromosomes, the stability of chromosomes in yeast. And I'm a very strong advocate in smart people can be trained in lots of different ways, but you can't train them to be smart. And so I've been tremendously blessed with having Sabrina here as part of the research core. I'm not down here all the time, but Sabrina is. And I couldn't have, the research couldn't have progressed this far without her. So with that, I'll hand you over to Sabrina.
Thank you, Simon. So typically with these talks, we like to end them with the thank yous and the acknowledgements, but given the purpose of today, I'd like to start with the thank yous. I'd like to thank everyone on the Murdoch study staff, from Carla to Leah to Mickey to Sarah to Perla to, I'm sure I forgot a name right there, but all these people, which predominantly ladies that I've interacted with, have been so dedicated and so helpful and so wonderful in getting these studies completed. So I wanted to say thank you. And most of them are right over there. I would like to thank the participants. Again, as Simon said, without the people that believe in our research, Leah, I forgot Leah. Without the participants who believe in our research, you would have never taken the time out of your day, out of your life, and shared your personal information with us, and as someone was saying earlier, your body parts, and participated in these studies. And so without you, we couldn't have done what we've done and what we're going to do in the future. So again, thank you. And I would like to thank Mr. Herman Stone and his family. Justin is here today, and also Jason Cox, who just gave such a wonderful speech. Again, much of the research today that I'm presenting was supported by them. And again, it would not be possible without them. So thank you. So we all know that MS is a complicated disease, it's a complex disease. In talking with groups of people with MS, one of the main themes that comes out is this very individualized disease. Every person's experience with it is different. So there's a wide array of symptoms that a person can experience depending on where in the brain and the spinal column the lesions occur. It's a disease of complex progression. So there's 3 main disease types. So we have relapsing remitting, where the disease progresses through periods of relapse and remission. There's secondary progressive, where at some point people that start out as relapsing remitting, they transition to a progressive form of the disease, where you don't get the relapses anymore and you just have steady disease progression. And then there's primary progressive, which is the most aggressive form of the disease, and thankfully the least common, in which from the very early presentation of symptoms, the disease continues to progress without remissions. And so we tend to present this as a really nice, tidy chart, but in reality it can be a little bit more messy than this in terms of people being able to identify when is an attack occurring, when is an attack not occurring. I know a lot of people that when they were diagnosed, they go back in hindsight and they think, oh, that one time that I had, my fingers went numb for a month or something, that might have been MS, but it went away, and so I didn't get diagnosed then. And so it can be a lot more messy history than that. It also has complex origins. So we know that there's a genetic component to MS, but MS is not one of those diseases where a single gene is responsible for it. And in fact, as Meredith introduced earlier, we have these genetic variants that everybody has. There's over approximately 200 genetic variants that the MS research community has identified that increase MS risk. But this isn't destiny. So a person can have, multiple of these risk variants, but that doesn't mean that they're going to get MS. It just means that their possibility of getting MS goes up. And so we know that people with a genetic predisposition to it get acted on by environmental factors. So things like viral infections, like with Epstein-Barr virus, smoking increases MS risk. And there's probably a whole host of factors that we haven't identified yet that also probably increase someone's risk of getting MS. And so what we have is this complicated situation where we have people that have different genetic predisposition to developing MS, that they're then acted on by environmental triggers or environmental risks. And of those that develop the disease, they have complex symptomology. So people are going to present differently. Some people are going to have trouble with their vision. Some might suddenly have trouble walking. But they all have the same disease. And what this kind of boils down to is diagnosis can be very complex for MS. And so currently for diagnosis, what we really want to do is rule out other diagnoses, and the gold standard being the MRI, like Simon was talking about earlier. And we want to know that these lesions are attributable to MS and not an earlier injury or another infection or some other neurological disorder. And so what they really want to see is that evidence that damage is occurred in at least 2 separate spots in the CNS at separate points in time. And so MRI imaging is difficult, and it's expensive. It's usually something people do annually, and it's not really ideal for diagnosing, and it's not very ideal for determining whether a particular drug treatment is working or not. And so ideally what we want to do is kind of untangle the complexity and develop ways to have improved diagnosis, better targets for therapy, improved treatment, better disease management, and being able to determine ways to predict whether a drug is working or not, or whether someone's disease is going to progress or not. So I stole a couple slides from Simon. If you're going to steal from the best. So, hey boss. So we want to develop biomarkers with the Murdoch MS study. So we want biological markers that will tell us diagnosis and prognosis. And as Dr. Bostrom and Dr. Nagy talked about earlier, they developed these omics profiles for us, which we're then applying a systems biology approach to. So we're combining genetic information, metabolomic information, gene expression information to try and unravel the complexity of MS story. And so we're developing these signatures of MS. And as he said previously, we're looking at these different layers of biological information, so DNA, RNA, proteins. And the study of the entire DNA complement in a person is the entire DNA complement in a person is their genome. The entire RNA complement is their transcriptome. And the study of these are the omics. So the study of genomes is genomics, study of transcriptome, transcriptomics. And so this is representative of the type of omics research that we've been doing and applying to the Murdoch study samples over the last couple years. So to start out with, I'm going to start out at the very base level, genetic information, DNA, and that is the genomics study. And so we have the central cohort out of the approximately 1,000 people that we've been looking at in the Murdoch MS studies that are very special to us because they're a very good starting point. These people are non-smokers, so we know smoking can affect the immune system. We know it can affect MS risk. And so we want to be able to eliminate that as a factor that might confuse some of our results. They're not receiving drug treatment. So you expect that when you're getting a drug that's treating MS, you would expect it to affect systems of the body that are relevant to MS. And so we want to make sure that what we're detecting, what we're seeing is related to MS and not to a drug treatment. And we have them matched with controls that are age, sex, and race matched, and in some cases BMI matched. So match them up as carefully as we possibly can. Try and eliminate as many variables as possible. So we know people are very complex and lots of variables in their lives, and we can't control for environment and everything in the same way that we can with a mouse. So match them up with controls, and this is what Meredith's lab has done for us. Isolate DNA from blood samples, and then look for these genetic variants. And so we've done some of these where we've looked at all the 200, or we're in the process of looking at all 200 genetic variants. We looked at an initial subset, and we're in the process of hopefully doing the rest of them. And ideally, we'd love to do the entire cohort of 1,000 people and get all 200 risk factors genotyped. The next study that we've looked at is metabolomics, and so this is primarily done by Dr. Winicky over here in Dr. Nagy's lab. And again, we started out looking at this base central cohort of people within the main group. that are non-smokers and not receiving drug treatment. And so Jason and Kevin developed these metabolic profiles, so looking at all the levels of small molecules in the blood, so vitamins, amino acids, sugars, that sort of thing. And what we got out is these models. So we want to know which metabolites are different in people with MS than in people that don't have MS. So in females, they developed a model of 124 metabolites that were different in people with MS, or females with MS and females without MS. Unfortunately, we have to do a lot of statistical tests to validate these models, and so it didn't pass the statistical validation model. It did pass it for males, though. So in males, we've got a collection of about 136 metabolites that can differentiate between people who have MS and people who don't. And so this is just kind of a visual representation of that, where each blue dot is a person who acted as a control in our study, and each red dot is representative of a person who had MS in our study. And these 136 metabolites, or 100 some, this is actually the entire male and female cohort, can separate out and predict who has MS versus who doesn't. And so what this translates into is these metabolic pathways. So we can have multiple metabolites in each metabolic pathway, and we can say, okay, we know that this metabolic pathway is probably implicated in some way in MS, in the pathology, in the progression in some way. And it's actually a very complicated thing, because we don't just have one metabolic pathway, we've got dozens of metabolic pathways that can be implicated